This is a replication of the Watson and Crick DNA model. As you can see, it has a double helix strand or a parental strand that connects to the bases. These are the nucleotides. The green is the deoxyribose sugar, white represents phosphate, and for the nitrogenous bases, red represents adenine. Yellow is thymine, purple is guanine, and blue is cytosine. There are only two combinations of nucleotides. Adenine pairs with thymine, cytosine pairs with guanine, and these are the only two combinations you can have. Replication begins with the double-stranded DNA being separated, and each original strand, called the parental strand, is used as a template for the complementary base pairing of nucleotides to make new molecules. The enzyme that makes this process occur is called the helicase. The helicase disrupts or weakens the hydrogen bonds connecting the bases of the nucleotides on the strands. This then exposes the nucleotide bases. Each separate strand of DNA molecule acts as a template for the production of a new strand of DNA. DNA replication occurs in the 5' to 3' direction, adding new nucleotides to the end of the 3' newly forming strand. This is the 5' end and the 3'. On the other side, this is the 5' and the 3' end. Next step of replication process would be adding nucleotides against each strand like this. DNA polymerase acts as a glue and attaches the pieces. It picks up free nucleotide units floating in the liquid background of the nucleus and inserts them opposite their complementary base partner, using existing strands as templates. The last step is to backtrack to proofread and edit the strand. DNA polymerase 1 does this and corrects any base pair errors by splicing out the incorrect base that was inserted into the new strand and replacing it with the correct one. Helicase goes through and unzips the rest of the DNA, and the other enzymes work hard to finish building the cell. Then an enzyme called ligase seals the strand together, and the final base pairing is checked by another DNA polymerase enzyme. This is what cell replication looks like after it went through the process.